In this video, I'll hopefully explain to you the cell cycle and the major ways in which we grow and divide and are able to reproduce. The processes I will be focusing on are mitosis and meiosis. The cell cycle is the overall life of a cell, like the one on the left, from the time it is created to the formation of its daughter cells, the two identical cells on the right. To more easily understand division processes, we will examine them on the level of humans through this rather menacing muscle man. This guy is comprised of thousands of cells that make up his blood, tissues, organs, and overall body structure. These cells also all work together to carry out processes that help the muscle man survive. But how are all these cells created, and how do they all carry the same DNA? In the human life cycle, cells are first created through the fusion of haploids, or sex cells, that is, sperm and egg cells. Each sperm and egg cell has one set of 23 chromosomes. When fertilized, the egg becomes a diploid, a cell with two sets of 23 chromosomes, or 46 chromosomes in all. It is then through a process called mitosis that this zygote, the fertilized egg, will grow and develop into a child with the creation of somatic cells. Somatic cells are all the cells of our body except the reproductive cells. Let's look closely at a cell undergoing mitosis to see more in depth how the process ensures that a complete and identical genome of 46 chromosomes will be present in daughter somatic cells. In this simulation, the process of mitosis is shown. First, we'll watch it in full, and then we will slow it down to go through it more in depth. The mitotic process begins with interphase. This is when the chromosomes duplicate and the cell prepares for cell division. There are three different stages of interphase. In the first stage, G1, or the first gap phase, the cell grows. Then, in the synthesis phase, the cell continues to grow as chromosomes are copied. Finally, in the G2 phase, or second gap phase, the cell grows more as it completes preparation for cell division. Prophase is the next step, where chromatin fibers become tightly coiled, nucleoli disappear, and chromosomes appear as two sister chromatids. Then, in prometaphase, the chromosomes become more condensed and spread in the nuclear area. In metaphase, centrosomes form at opposite poles, and the chromosomes align across the center of the mitotic spindle at what is called the metaphase plate. Anaphase then allows the sister chromosomes to part and become a full chromosome. These move towards opposite sides of the cell, giving both poles full and complete identical sets of chromosomes. In the last step, telophase, daughter nuclei form in the cell and a new nuclear envelope begins to form. In an additional step, called cytokinesis, the cytoplasm is pinched into two through a process called cleavage. A cleavage furrow, marked at this point right here, is formed. It is at this time that the complete creation of two identical daughter cells is finished. During interphase of mitosis, DNA replicates to ensure that processes are continued in the cell cycle and that there is enough DNA to create duplicated cells. If this doesn't occur, and the mitotic process spirals out of control, it may cause cancer, characterized by rapid and potentially harmful cell division like is taking place here. The cell cycle and mitosis are regulated via checkpoints. Checkpoints are just control points in the cell cycle where start and stop signals regulate occurrences in the cell. These signals are regulated via signaling molecules in the cytoplasm that are usually protein kinases or cyclins. When a cell reaches a certain checkpoint and receives a go-ahead signal, the cell is able to continue on the cell cycle. If it does not receive this signal, the cell exits the cell cycle and enters a non-dividing state. Cyclins allow the kinases to be active, creating cyclin-dependent kinases, or CDKs. MPF is a CDK and triggers a cell's passage into metaphase. So you might be wondering at this point then, if mitosis creates all our body cells and makes them identical, how are we all not the same? How does each and every one of us have a distinct look, distinct traits that differ from organisms in our same species? Well, like I said at the beginning of the video, zygotes, or fertilized eggs, are created through the fusion of a sperm and egg that each only have 23 chromosomes. These cells are called gametes and are created through a process called meiosis. This process creates cells that differ genetically from the parent cell in each other and reduces the number of chromosome sets to 23. If these cells were instead created through mitosis, chromosome count would continue to double from generation to generation. Meiosis instead maintains a constant number of chromosomes in a species from generation to generation. The process does so by beginning with a duplication of chromosomes and then going through meiosis 1 to separate the homologous chromosomes and later to meiosis 2 to separate the sister chromatids. The process ultimately creates 4 unique daughter cells with only one set of chromosomes each. Mitosis and meiosis are very similar processes in that they both have to do with cell division and go through the same phases. However, what distinguishes them is that meiosis creates haploid cells like sperm and eggs that can join to develop a zygote and go through the mitotic process to grow and develop. During the process of meiosis, a special process called crossing over occurs. In this process, cohesions hold sister chromatids together while the maternal and paternal DNA strands are broken at core bonding points. 2 homologous chromosomes are then attached together and each end of the break joins to the corresponding segment of a non-sister chromosome. Together, this creates a crossover that is completed as the chromosome condenses more. All in all, meiosis ensures genetic variation as it creates 4 haploid cells that differ due to the events of crossing over. In making connections with random parts of both paternal and maternal DNA, each gamete cell is different, meaning that all of us are genetically unique and different from one another. The process, however, is continual, and once we pass on our gamete cells to offspring, they have the ability to grow and develop their cells through mitosis and create gametes through meiosis, 2 very important cell division processes. Thanks for watching. Hopefully that was kind of helpful.